Thank you so much, Dennis, for inviting me to give a talk at the lunch hours. And hi, everyone. Thanks for joining. Uh, so I'll tell a little bit about myself. So my name is Bhavin Patel. I'm a senior manager in a mass spec reagents group at Thermo-Fisher Scientific. We are part of the bioscience division. We work closely with our instrument division team as well, who builds the mass spec. So our group here is the mass spec reagents group. We have 3 different focus areas, and now we started another one, fourth, where we look at the standard calibrants for mass spectrometry. We have sample prep focus area, and the quantitative reagents is the third one. And more recently, last couple of years, we have been working in a structural biology area as well. So I've been with the Thermo for about 11 years now. I started in R&D as a senior scientist, and last 2 years I've been managing the team here for the mass spec reagents group. So I'll tell you more about what we have been doing in last couple of years, and more recently what different products that we have commercialized. And feel free to ask questions in between. This is meant to be more interactive, so and I will keep the chat box open here. So if I see something, I can ask that. But feel free to interrupt and ask questions. So in agenda, I'm going to talk about the sample prep area where we have been working on, we commercialized EasyPrep technology 6 years ago. And then we have kind of building upon that with the different formats. More recently, we just commercialized last month the magnetic bead-based sample prep method. And we also have developed the automated sample prep platform called Accelerom in collaboration with our CMD instrument division team. TMT is one of our key product area, as we heard this morning from a lot of the from Dennis and Stephanie. This is something that has been with this group since it was commercialized in 2008, and we have been kind of building upon that portfolio. So I'll talk more about what we have been doing with the TMT Pro. We have developed the solution stabilized in a 96-well plate format, and how we can actually adopt this, for the better throughput workflows. And another focus area for us in quantitative reagents is the custom peptides. So this is something that we have been support through our mass spec reagents group. We do have a custom peptide synthesis services, and we do have done some lot of work around development and validation of targeted mass spec assays using this custom heavy label peptides. And we also have commercialized some of the kits and reagents in that area as well. So when we start out with kind of thinking about what is the end goals of proteomics researchers, researchers like to identify, quantify, and characterize the protein of interest. And there are a lot of different technologies out there, Western blot being the gold standard method, ELISA, Luminex. And when it comes to protein quantitation, mass spectrometry really plays a bigger role here. And also mass spectrometry helps with the cryo-EM technology to also provide more structural characterization of the protein as well. So in the proteomics, this is kind of how we look at it. You have 2 workflows that we look at. On the left-hand side is the discovery proteomics, and on the right-hand side is the targeted proteomics. So when we think about discovery proteomics, you pretty much start with the whole cell proteome. You do the trypsin digestion, you have tryptic peptides, and you do the shotgun experiments. And this method, what we do in a discovery proteomics is looking at the global proteome identification. And here, now you can see about 1,000 to 10,000 of different proteins that are present in your sample, and it depends on a sample type and complexity of the sample. What you get in the end is a relative quantitation of the entire proteome. Targeted proteomics has a very different questions that we are asking. We are only looking for protein of interest in the complex samples. You still do the same mass spec sample prep method, but your instrument now, that method you use is more like a targeted experiment, where you are actually looking at develop the targeted assay, and you can get accurate and precise quantitation of selected group of proteins. With this method, typically done in the tens and hundreds, we have developed a SureQuant method and commercialized that a few years ago that allows you to go even in 1,000 targeted quantitation. Even with the discovery and targeted proteomics, the sample prep is similar. So as we see here, we have a lot of different reagents that we have developed over the years. EasyPep is what I'm going to talk more about, but we also have IP to mass spec kits. We have fractionation kit, phosphopeptide enrichment kit. For plasma proteomics workflow, we have abundant protein depletion kits, C18 cleanup columns, and peptide desalting columns. So regardless of which proteomics method you're using, the sample prep is going to be similar. When we look at the quantitation, goals are different, however. For discovery proteomics, we will do label-free quantitation using DDA-DIA method, or you can do SILAC, TMT, TMT-Pro reagents. For targeted method, the short count assays, SRM, PRM methods, using the heavy isotope label peptide. So I'll start out with the sample preparation focus area that we have been working for many, many years. We used to have a kit that we commercialized when I joined the group for cell culture based samples. And that is what the traditional workflow would look like. You start with the lysing your cells. You do reduction alkylation in a sequential fashion. You may have to do the protein cleanup before you do the protein digestion, and then you do the peptide cleanup. And that takes about 50 steps and 2 days to prepare your samples before you can do the mass spec analysis. Some of the pain points with this traditional workflow is you get incomplete sample lysis, your reduction alkylation and digestion is not reproducible, you have the sample loss along the way. Also, the reagent quality plays a big role. So if you're not using high quality reagents, it can also introduce variability. And the biggest one is a lot of manual as well as number of steps. So what we looked at kind of each and every step and try to further optimize them. And there are a lot of other groups has also done the similar thing. With the EasyPep mass spec sample prep kit workflow, we actually improved the lysis by combining and removing the sonication steps. So if you have a cells or tissue, you basically use the EasyPep lysis buffer and add the nuclease to the lysis buffer. So then you don't have to do the sonication. And we combine the reduction alkylation. So you can actually just do the one quick 95-degree for 5 minutes, and you have your samples ready for the digestion step. And with the protein digestion, we combine the two enzymes. So trypsin is most commonly used. We combine that with lyc, and now you can achieve the digestion in 1 to 3 hours. And once you have this digested sample, you do the cleanup at the peptide level, and that reduces down the total time to about 2 to 4 hours. And the number of steps are now reduced to 24 steps. So if you look at this workflow in terms of how it breaks down, and the conventional workflow with the multiple steps, it's difficult, time consuming, and it's tedious. And on the right-hand side is what we have developed the solution for is the EasyPep technology, which is adding the nucleus to the lysis buffer, combining reduction alkylation in 10 minutes, digestion and cleanup. And all the reagents to perform all these steps are included in a kit. So this is simple, fast, and convenient method. So initially, we started working on this technology. We worked with this spin column format. So you typically start with plasma, cells, or tissue samples, and you actually use these spin columns to clean up your digest. And as you see here, we kind of built upon that from spin column format to the 96-well plate format. And we also developed the EasyPep Maxi kit. And this allows you to prepare the samples from 1 microgram all the way to 2 milligram of static material. And there are different applications that researchers would be interested in trying different format because you have high throughput customers, you have customers who wants to do the phosphopeptide enrichment that requires a larger starting amount. We were able to kind of create the solution that accommodates for all different applications. So whether it's a label-free quant or short quant assays, TMT reagent labeling, or phosphopeptide enrichment. On the right-hand side, where you can see here is that with even the different format, the chemistry is the similar. It's just the format for cleanup is different. You are getting, this is example of HeLa cell line um, that are performed using different formats. You have mini spin column versus 96 well plate and the maxi columns. And we get about the similar number of protein identified, regardless of what format you're using. If you look at the peptide IDs, you're also getting about similar number of peptides. Now, I would say that this some of this data then when we are generating, these are run on a more like a older Q exactive instruments. So your numbers are not really what you will typically see with the more newer instruments. When we take these samples and run on a newer one, your numbers are getting close to like 6 to 7,000 protein identified. With this method, because we combine the trypsin and lysine together, you can get the percent zero miscleavages rate. It's kind of assessing your digestion efficiency greater than 90%. So with all the different formats, we are achieving close to 93% zero miscleavage peptide. So this chemistry that we developed is scalable, and it keeps the performance for all different formats really good. And even here, this says 10 to 2,000 microgram sample amount. We have developed the micro kit. This actually accommodates for 1 to 10 microgram in a 96 well plate format, and I'll talk a little bit about that in a few minutes. So how does this easy PEP sample prep workflow is designed? So there are two-stage process. The first one is you are doing chemical and enzymatic sample processing. So you start out with the cell palette, you lyse your cells, and then your lyse solution and extracted proteins, you reduce and alkylate. And this is a combined step, as I mentioned. So now you are generating reduced alkylated proteins plus the contaminant that are coming from lysis buffer, your cells or samples, and reduction alkylation reagents. And then you perform the digestion using trypsin lysine, and then you end up with peptides plus contaminant. Now there is always been this standardized workflow for mass spec-based proteomics sample prep method. And we looked at different chemistries to understand how efficiently we can clean up most of this contaminant so that they are below the noise level when we actually put it in a mass spec. And we, of course, had a lot of C18-based SPE methods that is already out there. But we started looking at the different chemistries to really compare them side by side. And we identified a chemistry that is actually more like a mixed-more resin-based, where you apply these peptides and contaminants onto these spin columns. So these are resin that are packed into the spin columns in a slurry. You remove the buffer, and then you apply your sample. Once you apply your samples and you do the binding step, and then you actually wash these peptides and contaminants with wash A, you will remove the hydrophilic contaminant. Then you wash the peptides and other contaminants with wash B, and then you will remove the hydrophobic contaminant. So having this mixed mode capabilities allows you to remove a lot of the contaminants in a same sequential fashion, and then you elute your peptides into the clean solution, and you dry them down, and you reconstitute into the sample loading buffer. So there are more proteins we were able to identify with the 30% fewer manipulation and 60% less time than our original current kit that we developed maybe like 15, 16 years ago. So how does this easy prep workflow will fit with the TMT labeling? Because this is something that we wanted to kind of show that you prepare your digest samples, you can use it for label-free application, but can you also, is this compatible with the TMT labeling downstream? So again, we are following the same method here. In this example, we have plasma, which is depleted, and then you dry them down, and then you dry depleted plasma samples. You use the easy peplysis buffer, you solubilize the protein, and you create the peptides with the contaminant. And these are your digested peptides. And then you load them onto the spin column to clean all the contaminants. So one of the idea here is, can we clean the TMT efficiently? So you can do the labeling with the TMT tag, either right after you do digestion and have that labeling of each peptide done using the TMT tag, or you can do the TMT labeling after you do the cleanup. Now, there are pros and cons of using this method. If you do the labeling before you do the cleanup, that allows you to remove the TMT during that cleanup step. If you, let's say, do after you clean up your peptide, then you can remove the excess TMT during the steps of high pH reverse phase fractionation. And that both approaches are valid. One key thing about TMT labeling at the peptide level, we do recommend doing the peptide assay because you really want to normalize your sample before you do the TMT labeling. Or if you have done the TMT labeling before you mix them, you can do the peptide assay, know the numbers, normalize them, and then mix them together at the same concentration. So this is an example shows that how EasyPep cleanup method removes the excess TMT. On a top graph where we actually are using the peptide desalting column, and you can see the lot of excess TMT is coming out in a very front end of the gradient. And then if we use the EasyPep column to remove the excess TMT, you remove the most of those excess TMT during that cleanup step. So it's very significant decrease in the excess TMT. It does a very good job of cleaning up the TMT using this mixed mode EasyPep resin. When we look at the EasyPep kit with our conventional workflow, you are getting better number of protein and peptide identified, as well as you're getting more better TMT labeling as well. So EasyPep cleanup removes excess TMT more efficiently than other peptide desalting methods. So as I mentioned, as we were kind of started developing this workflow from 10 to 100 microgram, there was a lot of interest, specifically in a sample limited scenario. If you're working with like clinical samples like FAPE blocks, or you have biopsy samples or shortage cells, like few number of cells, or depleted plasma where you don't have a lot of starting material. That was one of the customer needs that we wanted to address. So we looked at our chemistry and say, okay, can we scale down the current reagents to accommodate for 1 to 10 microgram of starting material? So we took the HeLa S3 cell lysate, and in this example, 1 microgram, 5 microgram, or 10 microgram of starting material. We are seeing very good number, equal number of protein identified, about 4,400, and then the peptides around 29,000. And when we look at the % zero miscleavages, we are able to maintain that greater than 90% zero miscleavages rate. So we are consistently seeing close to 93%, regardless whether you have 1 microgram starting material, 5 or 10 microgram. And we are keeping the % reduction alkylation efficiency close to 99% or greater. So this really standardized workflow enables the more efficient sample prep for 1 to 10 microgram sample amount in less than 3 hours, and yielding better protein and peptide IDs with the lower miscleavage rates. One thing I would say that a lot of times when you have 10 microgram, you can do the peptide assay, you can measure the amount, but as you go close to like 1 microgram, there's not a really good way right now to know exactly what your yield is. So because you don't have enough material to really understand. So the approach that we have been using and recommending is you just run different concentrations, like 200 nanogram, 100, 200, 500, 1 microgram, and look at the TIC area, and then see how where that matches with your actual run when you do it. So while we were developing this EasyPep chemistry, we had a program that was looking at automating this workflow onto a robotic system. So we worked with our CMD group and developed this Accelerome sample prep platform that's shown on the left-hand side here. So this is an enclosed system with the software and everything is locked in. And it's the idea is to automate the liquid handler in a way that actually works in a much more simplified manner. If you have seen like some of the Hamilton robots or Pekin robots, it's a much more complex assembly to set up the methods and everything. This is actually all been locked in. And then we develop the reagents, and we are using the same EasyPep chemistry uh, to perform the whole sample prep. And one thing that we wanted to kind of look at it while we were developing is how are we going to quantify the sample amount. So we actually add in the UV cell inside the system. So it actually allows you to quantify exactly how much material you have before you can do the TMT labeling. And we have developed a different kit solution that works on this robot. So we have a label-free sample preparation kit, TMT 11-plex sample preparation kit, as well as the 16-plex sample preparation kit. So this is 16-plex is with the TMT Pro reagents. And these are ready-to-use kits that requires very minimum hands-on time to set up the experiment. So it's really very user-friendly. You have the software that is inbuilt onto the system. You don't need any computer or anything. And then you pretty much set up your kit, which kit you want to use. You bring out the reagents from the kit. You put it in the right locations onto the instrument deck, and you pretty much walk away. So this is a very robust and intuitive software that supports the scientists in their journey from experimental design through the data analysis. So it's a complete workflow solution for proteomics mass spec sample preparation. So if you look at a little bit more how the acceleromes works, on the top side, you start again with the protein extracts. You do the protein quant that is using the UV cell that is inbuilt. You load samples and kit component, reduction, alkylation, digestion, and you dry constitute and do the LCMS analysis. So we wanted to compare manually when we do EasyPep, how would that compare with Accelerome? And as you can see here, when we look at side by side, we are seeing very comparable number of unique peptides identified, about 33,000, and very equal amount of protein groups that are identified, close to 4,800. And when we look at the other characteristics in terms of the sample preparation, zero miscleavages and reduction alkylation is very similar. We are seeing slightly better percent zero miscleavage rate when we run this on our Accelerome system. So very comparable identification matrices with improved digestion efficiency using the Accelerome sample prep platform. As I talked about, this platform allows you to also do the TMT multiplexing, and you don't have to really do any hands-on work. So we wanted to compare what sort of labeling efficiency we are getting. In this particular case, as you can see here, we are getting very close to 99.8 or 7 percentage of the TMT labeling. We don't see a lot of non-labeled peptides. The zero miscleavage is consistently coming out close to 98%, and reduction alkylation at 99%. So we are getting really good labeling efficiency as well as the mass spec sample prep quality attributes. Another experiment we did using the system is where we actually do 16 TMT Pro 16-plex. So we take 30 microgram of starting material. We have 8 different replicates. Each sample is labeled with individual tag. And then you have 60 microgram of starting material, and we label them with the other set of 8 TMT Pro individual tags. And as you can see here, when we look at the peptide abundance, we are getting very good numbers for the peptide abundance, and you can see the difference of 30 to 60 microgram. When we look at the CVs for 30 micrograms, 60 micrograms, we are getting CVs close to 10%. And when we look at the ratio of 60 microgram over 30 microgram, you are getting ratio close to 2.0. So that kind of matches with what how we are starting the material. The expected ratio is spot on. And this is even we are getting this kind of quantitation without doing any sort of normalization of the data. So this automated sample prep with the TMT Pro labeling really allows you to get accurate and precise quantitation. It is cost-effective solution for the functional proteomics. So the next topic I want to discuss about is more recent launch that we have been working on for the last few years. We kind of looked at a lot of different beads that are commercially available, as many of you working in a proteomics area know about SP2, SP3 methods. And there is a workflow that has been put out there, and customers are using it. So we wanted to kind of look at that very closely. So we are part of the group that I'm here with is part of the legacy peer side. I don't know if you're familiar with the boxes with the handprints on them. Uh, this particular site is known for protein biology reagents, and we have a lot of bead chemistries that we develop in-house, cross-linkers, um, as well as some of the Western blot solution as well. So we wanted to kind of look at the commercially available magnetic beads and see whether we can actually develop something that would be better than what's out there. So we spent first 2 years really just screening, trying a lot of different beads, and we were able to identify the beads that we think is really good for this kind of applications. And these are the beads that are, we make this in-house here at the site. So what we have done with this one is kind of commercialize a little bit different flexibility for customers. So these are called EasyPrep Magnetic Mass Spec Sample Prep Kit. So we have 3 different options that we commercialize. The one is we can still do the EasyPrep digestion using the current reagents in an EasyPrep technology, where we do the lysis buffer reduction, alkylation, and digestion. The cleanup method, instead of using the mixed mode, you use these magnetic cleanup beads. And the binding and wash and elution buffers are also optimized for this workflow. So this is more like a SP2 method, where you make the peptides with contaminants and you clean up using the magnetic beads. So we have 96 reaction kit and the 20 reaction kit. We are also has commercialized the standalone beads. So we call this Proteomics magnetic cleanup beads. And the magnetic bead slurry is 25%. They come in a 5 ml size. And having this as a standalone allows you to do the SP3 method, which is basically you start with the protein aggregation capture at the protein level, and then you do the digestions on a bead, and then you get the sample that are ready for the mass spec analysis. So we wanted to give customer flexibility, whether they prefer to have the full kitted solution or they can actually use just the beads using the SP3 method. So here are some of the data comparing this magnetic versus the mixed mode resin EasyPrep method. When we look at the kit performances side by side, these are again taking the HeLa as a cell line, comparing the magnetic in a blue and the EasyPrep in an orange color. And when we look at the unique peptides, we are seeing very similar number of unique peptides and the protein groups using these both methods. When we look at the yield, and one thing I didn't mention in the yield for the EasyPrep, and this is something that was really a key product specifications when we were developing, is the yield. Because as many of you know, if you use C18, depends on a sample type you use, you may have very high variability during the cleanup step. And the yield could vary from 20% to like maybe 60%. On a different day, it could be different, right? So there's a lot of variability coming from the cleanup step. What we found with this mixed mode resin cleanup method with EasyPrep, we consistently get at least 50% yield. And in some of the cases, you may go up to 65 to 70% yield. And when we compare that with the magnetic bead, we are seeing very similar percentage of the peptide yield after the cleanup method. When we look at the digestion efficiency, we are getting very close to 96% digestion efficiency for the zero miscleavage peptides. And the reduction alkylation is very close to 99%. And since this is a magnetic bead-based workflow, you can actually automate that workflow for the cleanup part using the Kingfisher automation system. So we have a inbuilt, like the script that can be uploaded onto the platform, and you put the beads and all the other reagents, and you can automate the cleanup part in a 96 well plate format. When we look at the triplicate samples uh, analysis and look at the reproducibility, at the peptide level, we are seeing close to 89.4% common peptides. And at the protein level, that number goes to about 94.5%. And also, this magnetic bead format is compatible with the TMT workflow as well. So if you are not doing label-free, and if you want to do higher multiplexing, you can actually do the TMT or TMT Pro labeling. And we get a close to 99.6% TMT labeling efficiency. And the CVs are close to 6.3%. In this particular example, we wanted to label different samples, same samples with the different tags, and you can see the peptide abundance using the box plot graph looks really, really reproducible. So we get excellent kit performance with label-free as well as the TMT workflows and good compatibility with the automation system. Since I talked about SP2 and SP3 workflow, I wanted to kind of show some of the data that we have generated for this kit. So we compare the SP3 workflow using the same magnetic cleanup beads, and we also compare our EasyPep magnetic with SP3 and SP3 kit from a competitor. So the sample we are taking here is 50 microgram of the HeLa cell lysate. You prepare the samples using all these three different methods. So EasyPep magnetic is where you do the EasyPep digestion part and then do the cleanup using magnetic beads. SP3 is the pack method, and SP3 kit from the competitor following their recommended protocol. As you can see here, our EasyPep magnetic and SP3 workflow are very comparable. And we get more number of peptides identified. And when we look at the protein groups, we are getting very close to now 6,000 proteins using the both EasyPep magnetic and SP3 workflow. And it provides more number of protein as well compared to the competitors using the competitors SP3 kit. If we ask question about what's the overlap of proteins between the EasyPep magnetic, which is SP2 and SP3, we get about 93% of the protein that overlap between the two methods. And when we look at the competitors kit, we are getting about 88% overlap, but we do see more proteins identified using our EasyPep magnetic workflow. And when we look at just SP3 workflow, which is kind of more like-to-like comparison with the competitor, there is a 90% overlap, but then you get extra 500 proteins using this kit. And the CVs of the protein abundance across the board, we are seeing very good CVs that are with our workflow and kit, we are getting CVs that are close to 5% compared to 8% using the competitor's kit. So very comparable IDs between the EasyPep magnetic workflow, SP3 workflow, as well as using our proprietary magnetic cleanup beads. And we get more IDs than the competitor SP3 kit. This is kind of shows the example when we were looking at and developing these beads. We went through a lot of different iterations of the bead, and one of the key criteria we wanted to use is how these beads are compatible with the plastics and organic solvents and pH. And on the right-hand side, what you will see here is this is our beads when you actually have the tip comb, the bead by itself, when you rinse the bead, when you do the sample loading, you do washes and you do elution. And you can see that we don't have issue with the like other beads that are out there where the beads actually sticks to the plastic and you basically lose your sample and beads along the processing steps. So on the left-hand side, this is kind of what the layout looks like for the Kingfisher. plate setup. So you have a tip comb, you have a bead plate, you have a bead rinse plate, you have sample plate, wash, and elution. So it's just the 6 plates that you set up, and this is the workflow in a Kingfisher. So once you lay out all the samples, you hit a play, and it processes all the samples, and you get the eluted peptides that are clean, ready for mass spec analysis. One thing I would say, that method that we are using here for cleanup using the magnetic beads are more like a helix based. So you are doing high organic binding, and then you do the elution in a more aqueous buffer. So you don't have to do dry down. You may have to concentrate, depends on your sample loop size, but you should be able to directly inject the samples in a mass spec. And we also have a Hamilton system. So we have automated this EasyPep96 kit on a Hamilton system. So we also wanted to kind of create this full end-to-end workflow. So you do your digestion, you do your cleanup, everything is done, and TMT labeling onto the Hamilton system. So we have kind of updated our original script, including the TMT and the magnet onto the system. So if we use the Hamilton automated sample processing, we took 25 microgram and 50 microgram of HeLa, and we did the label-free quantitation. Here, when you run the 3 different replicates, you get about close to 6,000 proteins. 99% of them are very commonly seen between the replicates. So we see really good reproducibility between the replicates. And when we do TMT Pro quantitation, on the left-hand side, this is the TMT. And then you can see it's a really good peptide abundance. When we do TMT Pro at 25 and 50 microgram, we are getting the ratio of 2 as we expect. And the CVs are close to 10% or below. So we are really seeing excellent quantitation, precision, and accuracy for both label-free and the TMT-based workflow. And having the automated system is really enables you to do the end-to-end EasyPep magnetic workflow using the Hamilton system. So next few minutes, I'll switch the gears and talk more about the quantitative reagents. So as we heard this morning about TMT reagents, I will not go so much detail about how they work, but what we have more recently done, in terms of format. So as you are aware, there is a TMT and there is a TMT Pro product family. The TMT Pro supports more stable isotopes, so we can go up to 18 unique reporter ions versus the 11 plex for the TMT reagents. Regardless, they both are amino reactive reagents, so they are used for labeling the protein digest, irrespective of sequence or digestion enzyme specificity. So the workflow is very identical. So in this example, typical workflow, you have 18 different samples. You can create some good experimental design where you have replicates within them, and then you label with different tags. After digestion, you combine the sample, and then you analyze by LC-MS analysis. And these are typically done on a more of our tribrid instrument series. And then you do the data analysis using PD or MaxQuant software. So last couple of years, we have been kind of really looking into making a solution stabilized TMT Pro reagents. So these are basically, we spend a lot of time optimizing the stabilized solution. So it is a DMSO-based solution, but we looked at a lot of different formulation and found the one that is more stable, that we are utilizing in these different products that we commercialized last month. So you have TMT 10-plex reagents. These are 96 well plate format. You have 8 by 40 microgram. And then you have 11-plex in the same 8 by 40 microgram. 16-plex is in a 6 by 50 microgram. And then 18-plex in 4 by 50 microgram. And they are kind of PCR plate based with the different, as you can see here, each well is a different tag. So in looking at the performance of this format, when we take 1, 5, or 10 microgram of sample and label them using the solution stabilized TMT reagents, we are seeing very similar performance. You do get a little bit drop in a 1 microgram because we may be not loading about the same amount of material on a column. But we see very similar performance with the unique peptides and the labeling efficiency of 99% for 5 and 10 microgram. When you go down to 1 microgram, you are starting to see some challenges with the labeling efficiency. So you may have to kind of adjust the ratio, keep the volume very small because having the smaller volume actually helps to achieve a better labeling efficiency. So when we look at this solution stabilized TMT, TMT Pro reagents, we really wanted to maintain the activity in a stabilized solution. So this is a stabilized DMSO solution. The reagents activity we assure is 12 months at minus 20 degree. The activity preserves after 9 freeze-thaw cycles, so you can reuse the plate if you don't want to use the whole plate. These are developed and formulated using the validated automated filling equipments for manufacturing purpose. And it's also stabilized DMSO is compatible with the downstream workflows. When we look at the 96 well permit high throughput, and it's also easily compatible with the other automation platform as well. The foil that we put it on top is easily pierced and peeled. And there is a barcode on the PCR plate, allows you to make sure that you can identify and also helps with the orientation. And the plate has a rigid polycarbonate skirt in the bottom, and that is actually helps with the robotic handling system. And it's compatible with most of the automated liquid handling systems. And when we look at the performance, we have shown that it demonstrated more than 98% labeling of the protein digest. The performance between TMT and TMT Pro formats are very equivalent, and we can actually has ability to label small amounts of samples, like 1 to 10 microgram. So this is one of the applications that we did with this using this format and EasyPep kit. As I mentioned, there is a lot of interest in FAPE kind of samples. So we have optimized this workflow for removing the paraffin, removing the cross-linking, and we do that using our EasyPep lysis buffer directly. So it's basically, once you have this 6 different curls, we took like breast, colon, and lung, normal and tumor, and then we follow the EasyPep 96 micro mass spec sample prep kit. We were using 5 microgram of starting material. You do TMT Pro 18-plex labeling, you combine, and then you do the plate cleanup. You do the peptide assay to normalize them, and then do the high pH reverse phase fractionation to generate 6 fractions, and then do the LC-MS analysis. So if you look at this FAPE sample analysis, on the left-hand side is the PCA analysis. So we are seeing a really good grouping of like colon tumor, colon normal, lung tumor, breast tumor, and breast normal. So you can see it's really nice grouping of each sample type. We are seeing about 4,000 proteins and 26,000 peptides with a labeling efficiency of greater than 98%. And when we look at some of this differentiated regulated protein, when we look at some of the biomarkers, so in this case, ERBB2 is a known biomarker for breast cancer, is upregulated in only breast tumor sample. One of the biomarkers for colorectal cancer is CEACAM5, is upregulated twofold in a colon tumor samples. And coronin 1A is upregulated 2.5-fold in a lung tumor sample. So this really allows you to, having that standardized kits reagents and the multiplexing allows you to really get a really good biological differences with the high quantitation accuracy and precision. Any questions so far before I go into the targeted proteomics workflow?
It doesn't look like we have any questions at this time.
Okay. So I'll spend a few minutes on talk about the targeted proteomics workflow. This is something that we have been working for many years. And this is how we look at the targeted assay development workflow. Sample prep is, again, very critical steps there. You need to really understand your protein abundance so you can use the right sample prep method. The next thing is really generating the discovery proteomics data and utilizing that to select your peptide. And this is how we kind of recommend to develop the targeted assay. So if you have discovery data, you can actually select at least 5 peptides for each protein and make the crude peptide libraries. And then you screen them to determine the 1 or 2 best peptide that passes the criteria for synthesizing those peptides in aqua grade format. And I'll talk a little bit about what the differences between these two. But this allows you to really have a more success in the end, having the peptide that has a really good LOD, LLOQ, dynamic range, and the robustness. And in terms of once you have that peptide mixture that is validated, you can apply SRM, PRM, or short count method to do the targeted mass spec analysis. So the first step we recommend is using these peptide libraries to screen the candidate peptides. So from your discovery data, you select the 5 peptide at least per target. You get them in this crude peptide library format. So these are comes in. This is kind of a 96 plate format tubes. Each peptide is in 50% acetonitrile with 0.1% TFA, and you can get this peptide very quickly, as little as 2 weeks. And once you get this peptide, these are purified as crude, so there is no purification done on them. You can take the equivalent amount of peptide, mix them together, and then you run them, and then apply certain criteria. You want to look at the intensity of each peptide. You want to spike into your matrix and see heavy and light coelute. You want to avoid hydrophilic or hydrophobic peptide. You also wanted to see whether you can achieve the dynamic range, at least 2 orders of magnitude when you dilute the peptide down, and also make sure that you get at least 4 fragment ions, ideally with the Y ion series, because you are going to be using this peptide with a heavy isotope label on a lysine or arginine on a C-terminal side. And then once you have that selected peptide, then you can order them in aqua format. The cost of aqua is higher compared to the crude peptide. So that's why it's better to know these are the really good peptide to go with before you order them in aqua format. Aqua grade peptide follows very similar, the same peptide synthesis process using the AFMOC solid phase chemistry. But we purify the peptide using HPLC. Purity has to be greater than 95%. And we also do the amino acid analysis independently and with the precision less than 10%, and then do the quality checks using mass spec, tandem mass spec, and HPLC. We have 3 different kinds of peptide for aquagrade. You have basic, Quant Pro, and Ultimate. The real difference here is just how many times we do the amino acid analysis. So in Ultimate, you do 3 times, Quant Pro 2 times, and then basic is just 1 time. The delivery time for this aquagrade peptide is about 2 to 3 weeks using the express service, but the standard delivery is about 5 to 7 weeks. And we also provide the quality testing reagent results with that as well. One thing that no other vendor can is providing something what we call heavy light equivalency testing. So if you have a peptide that you are going to generate the standard curve using variable concentration of light and spiking in the constant heavy, we assure that both of this light and heavy has the amino acid content analysis and the LC-MS analysis in a peak area less than 15% from each other. So this is an example of the kit that we commercialized a few years ago for AKT-mTOR pathway quantitation. So this is a multiplex assay that monitors 12 different targets from AKT-mTOR pathway. And each peptide was synthesized in ultimate grade format. This is kind of the linearity that we see. We get at least more than 0.99 for most of the peptide. And LLOQ, which is the lower limit of quantitation we see, is close to, for most of the peptide, is 0.03 or 0.13 femtomoles on column. We also provide with these reagents ready to use Skyline template. So Skyline is a software that is most widely used for targeted experiments, and that also has a capability to generate the standard curve. So all you're doing is just bringing in your data, and it will do all the calibration curve as well as the determining the concentration from the unknown sample. As I mentioned, one of the methods that we developed on our tribrid instrument as well as Exploris 480 is called short quant method. If you do PRM or SRM based method, you have to know exactly what time the peptide is eluting so that you can schedule a window around that as you increase the number of peptide in your single analysis. With the short quant method, it is kind of internal standard trigger targeted method. So what you're doing here is you spiked in internal standard and you generate the library of each one of these heavy isotope labeled peptide. What instrument does in a real time is actually does the fast low res MSMS and then matches with the library for the fragment ion series. If it finds the match, it actually switches to generate the high quality MSMS data for both light and heavy. So in this case, you don't have to do any scheduling. All these things done in a real time during the peptide as they are eluting off. And you can do the data analysis in a Skyline. If you're interested, I'm happy to follow up on this method and provide more details. But this methodology is available on Exploris 480 and also all the tribrid instrument with the Tune 3.0 or higher. When we compare short quant with the PRM, what we see when you serially dilute your peptide, you are getting actually really good sensitivity. So we see 4 times higher sensitivity than PRM on the same instrument. And this is something that is really allows customer to deep dive and for the very low abundant targets, achieve that higher level of quantitation accuracy and precision. Also, it's maintaining the CVs close to 2.46% and the 2% for PRM. So really highly precise measurements about the LLOQ values can be achieved using the short quant acquisition method. I'm going to quickly show one applications for the phosphopeptide standard. So this is something we work with the SPRG group. They're part of ABRF many years ago. We supported some of the initial efforts to develop this multi-pathway phosphopeptide standard. And there are 131 phosphopeptide that cover 7 most widely studied signaling pathways. And this peptide mixture has been commercialized. And the way we utilize this standard, multi-pathway phosphopeptide standard, is you have your samples, you do EasyPep, you get the tryptic peptide digest, you spike in your phosphopeptide standard. These are heavy isotope level. And then you do the phosphopeptide enrichment. We also have a high select kit that allows you to do some magnetic bead based. And then you get enriched phosphopeptide with heavy isotope label peptide as well. And then you can do the short quant analysis. So this allows you to do like 131 phosphopeptide quantitation in a single mass spec run. When we were developing, we shared this standard to Professor Jesper Olsen at the University of Copenhagen, and they utilized the standard in a control with EGF stimulated for 10 minutes and 2 hours, IGF stimulation at 10 minutes and 2 hours. And you can clearly see here that this standard allows you to get much more precise and accurate quantitation at the different conditions. So they were able to see 103 phosphosites quantified using the Skyline. And you can really monitor this phosphorylation event with the more higher quantitative accuracy and precision. So I know we are at the time. Thank you for, I hope you enjoyed. I'm happy to answer any questions.
Any questions? Targeting guy? I mean, Mike, you have a question?
So hi, I'm Mike Kinter.
Oh, your thing's not on.
So I was just curious if you could explain, I guess I was confused about how the SureQuant could be more sensitive than PRM. As I understood it, it surveys and then switches into a PRM mode. So I was sort of struck by why it would be more sensitive than just PRM by itself.
I think it may have to do with the when you do the PRM measurements, you may be getting some more higher noise level when you do that. When you do SureQuant, you may be achieving that or removing that noise much more, because what your instrument is doing is you're gonna be really spending just time on that particular peptide all the time, right? So that's what I think, and I agree with you. When we saw the data coming out, it was like really striking difference between those two. But for some reason, the SureQuant acquisition method, even though it's doing the high-res PRM, is you are starting to see better signal to noise when you go down close to that LLOQ or above values.
Okay.
Thank you. Good information there. I think the DMSO solutions with the TMT reagents is something worth us following up on. I'm always, I should say, this is Rick Edmonds. I always wonder about cost, and so we're cost conscious, and so. Can you give a feel for cost of a few of these reagents? The magnetic beads, which I certainly appreciate, and the fact that they stick to plastic less is, for those who haven't done the experiment, may seem like a trivial fact, but that's a very real effect, and I appreciate that. But costs for, like, bulk reagents for the beads, and then maybe the 96 well easy prep kits, just ballpark costs.
Yeah, so the 96 well plate kit, we are looking at maybe close to somewhere between $15 to $20 per sample, I would say. This is the one downside of working in R&D is I don't have exact number, but I will definitely follow up with you. Cost is something that we always keep in mind when we are developing product, because as we know, that the field is growing, we are looking into how can we reduce the cost per sample, right? So with the magnetic bead, if you are buying kit, it's going to be slightly more, but I'm gonna quickly see if I can see the pricing on a website, and I can put the link here as well. But if you're getting 5 ml beads, it's like $320. If you are doing 20 reaction, you are looking at $550. So 96 is like $1,500. So we are close to about $15 per sample, I would say, for the kit format, because you're gonna have, I think the biggest driver when we see with the kit when we are making them is the trypsin lysine.
Sure. Yeah.
That's kind of like, everything else is not something that we can work with, but like, we can adjust, but the trypsin lysine is what drives the cost for a lot of these kit format reagents. But yeah, we are looking at somewhere around $15 to $20 for a reaction for both magnetic bead kit as well as the 96 well plate kit. But I'll definitely follow up with the more details about that.
I have one question. So in the initial part of your talk, you mentioned sample purification. So my sample processing involves sucrose gradient.
Mhm.
So can I use like this the fraction having the sucrose to purify using your kit?
So you are purifying the protein, right? In the beginning?
Yeah.
Yep. And then you want to do the sample prep on that purified protein.
Exactly.
Correct. Yeah,
Yeah.
that may be a little bit tricky. As long as you don't have any sort of like contaminant that will interfere with the digestion, you should be able to clean them up on a column format. using the EasyPep mixed mode resin. So you may have to look at the sample. So your sample is typically, is it too diluted or is it too, what is the concentration of your sample when you do the sucrose gradient?
Sorry. It's a sucrose cushion ranging from 10% to 50%. So
Okay.
I collect from somewhere between like 30% sucrose, you know, which captures monosomes and polysomes.
I would definitely follow up on this one with you.
Okay. Okay.
Yeah.
Yeah. I'll reach out to you. Thank you.
Yeah, what's your name? I just want to
Ram Shukla.
Ram Shukla. Okay. And yeah, if you have any questions afterwards as well, or any specific topic area that you maybe wanted to have a little bit more technical deep dive discussion, happy to follow up on those. We do have other reagents I didn't talk about on plasma depletion, phosphopeptide enrichment, IP mass spec kits. So any of those areas that if you see that we can discuss more on a technical side and see if that fits with what you need. Happy to do that.
If it's okay, Dr. Levin, I would, Patel, I will just share your email.
Yeah, absolutely. Yeah,
Okay.
absolutely.
Perfect.
Yeah.
Okay.
Yeah, feel free to share my email, and reach out to me, and I will we can follow up either by email or by the phone discussion.
Okay.
Can you also share his slides?
Yeah.
Excuse
me. Yeah.
His slides.
Oh, do we have a copy of your slides?
Yeah, I can send it out, Dennis, to you, and you can.
Okay, perfect. Thank you so much.
You're welcome.